Okay, first of all, I would like to say thank you to organizers to make me possibility to present my data here. In my lecture site, I try to show to complex between S100 beta and A beta peptides. We have in our lab this A beta version 140, not 142. It's like this region of our production. Generally, I'm represent the cinema community, so we interested in structure determination, protein solution. Why we interested in in this protein S100 beta? So first of all, it's quite small dimeric metal binding protein. This protein belonging to S100 family, these calcium binding proteins, which are usually dimeric. It's every subunit is contain these two calcium binding loops. This C-terminal loop. It's, let's say, canonic is 12 amino residues long, and N-terminal loop. It's means this quasi-F-hand, which is specific loop which contains 14 residues instead of 12. When they bind calcium, this shown it's quite big structural changes. One of these helices it changes orientation and opens these hydrophobic sequences. And here I show this complex S100 beta with some peptides there, TK12. The whole S100 family in humans, it contains 23 residues, but S100 beta is one of the best studied. To now, this is one of the most abundant soluble protein in brain. I think it's around 0.05% of all protein in brain. It is S100 beta. Yes. Also, why we are interested in this protein? It is long-standing interest in our institute. We have a lot of efforts to study this cell receptor, which name RAGE. It's receptor advanced glycosylated product, and this receptor responsible for transfer A beta peptides inside the cells. Also in cooperation with an NMDA receptor. And this is this complex S100 beta with RAGE. It was studied for it was studied a couple of groups. Here I shown this very old papers. It's 210 from Günther Fritz. It's shown this structure of RAGE receptor, which contains several domains. After that is transmembrane domain, it's a tizodolic domain. It is from this dates. This structure was prepared in our institute. It's mass spectrometry lab by Sasha Moisa 3 years ago, and you see that he showed that this S100 beta is from tetrameric structure which interact with three different domains. Here I show this three different views of structure which are based on different PDB structure available. This structure was solved by X-ray crystallography, and it's shown that S100 beta it's interact with different form of different domains of RAGE receptor. Nevertheless, it's the central part of this receptor. It's still available to transfer a beta peptides inside the cell. Here, this initial experimental data, which was collected by other group from Portugal, which are shown that A beta interacted strongly with S100 beta, but only in calcium binding form. In APA form, you see that it's on the proton-nitrogen correlation spectra. We have no changes at all, but after addition calcium, it's this form is named holo instead of APA, which is no calcium. You see that it's orange residuals. It's only a couple of these residuals which are available to detect on the spectra. This effect is still not clearly could be clearly expanded. I will discuss it a little bit later. We also have data that this theoflavine data which shown that under interact with S100 beta, S100 beta, it's strongly inhibited effect of fibrillation of A beta peptide. You see that this for 5 micromolar A beta, this we have strong effect of delay of fibrillation, and also this effect observed in both forms. It's without calcium and with calcium, but calcium addition it's changed the character of interaction between A beta and S100 beta. Additionally, later we have titration calcium binding form of A beta was titrated by A beta peptide, and this is shown this shown surface which are changes under A beta titration, and this possible for us. It is proposed model of 3D structure of such a complex between S100 beta and A beta. Next one, it is I show this electrostatic interaction between protein and peptides. You see that this electrostatic surface in case of S100 beta is mostly negative, and rather complete electrostatic surface of A beta peptides is completely different compared to S100 beta protein. You see this N-terminal of A beta peptides is located here. This is metal binding domain. Which comprise residuals between asparagine 1 and lysine 16. Also, we try to analyze it. What is changes in molecular dynamic processes in protein when we saturate S100 beta with A beta peptides. This is it's compare relaxation data which we collected in our NMR spectrometer for protein alone and in complex with A beta peptides. Analysis of this experimental data make us information about molecular dynamics and in three different structure in very low frequencies, in frequencies on nitrogen frequency. In our case, it's around 50 megahertz. And you see that this red one, it is data for peptide alone, and blue one, it is data for complex. And it is clearly visible that addition A-beta peptides to our protein, it is resulted in shift intensity of molecular dynamics in both on both frequency, on low frequency and on let's say middle frequency. It's time frame between microsecond and nanosecond. Here we are. I try to show what is residuals, which is changes under A-beta peptide binding. We will see that our data provides information that binding peptide provides this more decreased molecular mobility in residuals and in this helix in central hydrophobic core, which are formed these two helices. It means that it's A-beta peptides, which located here in the interface between subunits. It's provided this additional constraint for take this subunit together. Nevertheless, in this hinge region, so this more or less flexible loop, which connected to calcium binding motif together, we observe increased molecular dynamics in these residuals. But what this is it is hydrophobic interaction. So this is shown surface. It's hydrophobic surface for this structure, for this complex. And this two different view of this complex. Finally, we would like to see information about this complex, not from protein point of view, but from Alzheimer peptides point of view. So this data shown proton nitrogen correlation for N15 labeled peptides and unlabeled non-labeled protein. So we observe that many peaks from A-beta 140 peptides. It's not observed for this spectrum on this spectrum at all. So this is possible. It is two explanations, but both of them it's not true. One of them it means that this amide proton could be exchanged with water, but it is not true because we know that peptides it's deeper inside the protein. And another option that we have very strong oligomerization, the whole system, but experiments show that molecular mass of this complex it's not changed significantly from the S100 beta protein. It's also interesting idea if we see if we get close view for the structure of a beta peptide with S100 beta protein, we will see that its hydrophobic contact between peptides and protein is located only one side of this helices. It's I think it's clearly visible that from this side we have only glycine, so this is not hydrophobic sequence, not hydrophobic residuals located on this side of helices. And this fact we have to additional confirm it by experiments which we provide right now. And the last idea what we would like to check influence of metal nanoparticles. In this case, we have copper oxide nanoparticles with diameter 50 nanometer. And we would like to check if complex between S100 beta and A beta peptides formed under saturation with such nanoparticles, but it's at the moment we didn't have good sample, so we only check it is nanoparticles. It's influence the structure of S100 beta protein, and we see that it's really this nanoparticles could change a little bit structure of S100 beta protein in calcium loaded form. So this effect could be modulate of calcium binding affinity. And this conclusion, it is that S100 beta protein strongly interact with A beta peptides. We observed this changes, some changes in molecular dynamics properties under interaction with A beta peptides. And this different for inter-subunit interface we have decreased molecular dynamics, but for hinge region which increase molecular dynamics, and we have that nanoparticles could provide some structural changes and probably molecular dynamic processes also changes and interact with nanoparticles. Yeah? And this acknowledgement for for people in my group and in group in Poznan. This is all. Thank you.